Hello, everyone. We are going to cover mRNA vaccines in nucleic acid vaccines today. Nucleic acid vaccines have garnered a great deal of attention recently, with the successful launch of the SARS-CoV-2 mRNA vaccine setting the momentum for the nucleic acid vaccine platform. Because of the absolute advantage of these platforms in targeting highly variable pathogens, many vaccine candidates are in development, including SARS-CoV-2, influenza, and HIV. What is a nucleic acid vaccine? Nucleic acid vaccines use nucleic acid molecules as vaccine carriers. By injecting specific nucleic acid sequences of pathogens into human cells, the cells are prompted to produce antigenic proteins of the pathogens on their own, thus activating the host's immune system to produce protective immune responses. Next, I will briefly describe the innovations of nucleic acid vaccines compared to traditional vaccines. 1. Nucleic acid vaccines are simple and fast to develop and manufacture. They are easy to produce and can be produced quickly with the help of manufacturing in response to emerging epidemics. 2. The absence of infectious pathogens during the production of nucleic acid vaccines also eliminates concerns about incomplete virus inactivation or purification, ensuring their safety in humans. 3. High specificity. In addition, the genetic sequences of nucleic acid vaccines can be easily modified to target specific pathogens and diseases, adapting them to emerging infectious diseases and rapidly evolving pathogens, or to personalized immunity against heterologous diseases such as cancer. 4. In addition to safety and ease of production, nucleic acid vaccines have the ability to participate in antigen presentation to induce cellular and humoral responses in the host immune system. In contrast, conventional protein vaccines are less efficient at triggering CD8+ cytotoxic T cells, a class of T lymphocytes capable of directly killing infected or cancerous cells, responses. Let's first look at the history of mRNA vaccine development. The discovery of messenger RNA, mRNA, occurred in 1961. The concept of utilizing mRNA as a novel therapeutic agent emerged with the development of liposomal nanoparticles, LNPs, in 1989. The scientists demonstrated that mRNA packaged within liposomal nanoparticles could successfully transfect mRNA into a wide range of eukaryotic cells. This scientific breakthrough helped overcome the previous limitations of mRNA instability and high production costs, and led to substantial advances in mRNA delivery, manufacturing, safety, and synthetic modifications. With the 2019 coronavirus pandemic, mRNA vaccine research is expanding at an unprecedented pace and scale, with 2 COVID-19 mRNA vaccines receiving emergency use less than a year after clinical trials began. Next, let's look at the principle and working mechanism of mRNA vaccines. The working mechanism of mRNA vaccine includes the following 6 steps. 1. Design and synthesis of mRNAs. Key antigenic proteins on the surface of the pathogen are first identified and the corresponding mRNAs are synthesized based on the gene sequences of these proteins. The mRNAs are packaged in lipid nanoparticles, LNPs, to protect them from degradation by in vivo enzymes and to aid in their entry into the host cell. The LNPs enable the mRNAs to cross the cell membrane and enter the cytoplasm. 2. Expression of antigenic proteins. Once in the cytoplasm, the mRNA is translated using the host cell's ribosomes to produce antigenic proteins. This process is similar to the cell's own protein synthesis mechanism and is therefore very efficient. 3. Antigen processing and presentation. The generated antigenic proteins are cleaved into small fragments by intracellular processing mechanisms, and these fragments are captured by the major histocompatibility complex, MHC. 4. Activation of the immune system. T cells of the immune system begin to function when antigenic fragments are displayed on the cell surface. CD8+ T cells, cytotoxic T cells, recognize and bind to antigens displayed by MHC class I molecules and activate to kill infected cells directly. CD4+ T cells, helper T cells, recognize and bind to antigens displayed by MHC class II molecules and activate to help other immune cells, e.g. B cells and CD8+ T cells, to enhance the immune response. 5. Antibody production. B cells, with the help of CD4+ T cells, recognize antigens and begin to differentiate into plasma cells that produce large numbers of specific antibodies. 6. Formation of memory immunity. A portion of activated T cells and B cells differentiate into memory cells, which are capable of rapidly initiating a strong immune response when encountering the same pathogen in the future, thus providing long-term protection. Through the above mechanisms, mRNA vaccines are able to effectively mimic the natural process of infection and induce a strong immune response while avoiding causing disease. At the core of mRNA vaccines are RNA vaccine platform technologies, the application of which has significantly improved mRNA stability, translation efficiency, and immunogenicity control, laying the foundation for RNA vaccines in both therapeutic and prophylactic areas. The first major technology is structural modification. RNA is an unstable molecule that is easily degraded by extracellular RNA enzymes and was therefore initially considered unsuitable as a therapeutic agent. However, scientists have worked tirelessly to make mRNA vaccines a reality. Notably, in 2023, 2 American scientists used nucleotide modification, which allowed the synthesized mRNAs to evade the body's natural immune recognition system, significantly improve mRNA stability. They were awarded the 2023 Nobel Prize for this innovative contribution. This technology is key to mRNA vaccine technology, laying the foundation for effective mRNA therapies. No. Nucleotide modification. The main nucleotides used in mRNA vaccines are N1-methyl-pseudouridine and 5-methoxyuridine. Nucleotide modifications have the following effects. 1. Reduced immunogenicity. As just mentioned, nucleotide modifications such as N1-methyl-pseudouridine can change the folding of mRNA, reducing its chance of being recognized as a pathogenic RNA by the host's immune system. 2. Improved stability. 5-methoxyuridine alters the secondary structure of mRNA, increasing its resistance to nuclease and extending the half-life of mRNA. 3. Optimize translation efficiency. Improve the removal of translation byproducts, double-stranded RNA, dsRNA, reduce the interference of these byproducts with ribosomes and other translation machinery. 4. Enhanced protein yield. Through specific modifications, mRNA is more likely to bind to ribosomes and be translated efficiently. This image shows the structure of the mRNA, and we can see the approximate position of the fibrous cap and threaded tail. 1. Fibrous cap. The fibrous cap is a modification added to the fiber end of the mRNA molecule, usually 7-methylguanosine, M7G. The fibrous cap not only protects the mRNA from degradation, but also plays a key role in translation initiation. 2. 3-poly-A tail. The 3-poly-A tail is a sequence of adenylates added to the threaded end of the mRNA. The length and composition of the poly-A tail directly affects mRNA stability and translation efficiency. Nucleotide modification and optimization of the fibrous cap and threamer tail make the mRNA vaccine a safe and efficient vaccine platform. The second core technology, delivery optimization. Direct injection of mRNA into the organism faces a number of challenges. Therefore, naked RNA delivery is usually inefficient and difficult to achieve clinical applications. Figure keys A. Overview of nanoparticle RNA delivery. RNA must be encapsulated in nanoparticles and endocytosed into the cytoplasm. B. Polymer oligomer-based delivery. To overcome the limitations of naked RNA delivery, a variety of RNA vaccine delivery vectors have been explored. These vectors mainly include lipid nanoparticles, LNPs, cationic liposomes, and dendritic cell, DC, loaded RNA. These vectors can encapsulate and protect RNA and facilitate its entry into the cell, improving RNA stability and delivery efficiency. Among them, LNP has become the most popular and widely used RNA vaccine delivery system due to its superior ability to encapsulate and protect RNA. Lipid nanoparticles, LNPs, are nanoscale structures composed of a variety of components, the main components of which include ionizable cationic lipids, cholesterol, auxiliary phospholipids, and polyethylene glycolated lipids, PEG lipids. LNPs effectively protect mRNA from degradation by ribonuclease, RNase, in vitro and in vivo through their lipid bilayer structure. Second, The nanoscale size and charge characteristics of LNPs allow them to be efficiently taken up by cells through endocytosis. Finally, LNPs release cationic lipids in an acidic endosomal environment, which promotes the fusion of LNPs with endosomal membranes and the release of mRNA into the cytoplasm for translation. Schematic representation of the mechanism by which LNP-assisted mRNA delivery kills tumor cells via DC. The advantages are much, enhancing the delivery, stability, flexibility, and safety. Finally, let's talk briefly about immunomodulation. One of the technological platforms of RNA vaccines is to enhance the effectiveness of vaccines through immunomodulation, which is divided into two main components: adjuvants and immunomodulators. Adjuvants are substances added to the vaccine to enhance the immune response, while immunomodulators enhance the effectiveness of the vaccine by modulating the immune system's response. Okay, we've had a brief introduction to mRNA vaccines today. How was it? If you have questions, please leave them in the comments section. Make sure you subscribe and like this video. For more information, please visit our website. Have a great day. Bye.